Yes, it gives me, I'm Stephen Locarnini from the Victorian Infectious Diseases Reference Laboratory in Melbourne, and I'm giving a talk on the invitation of the APASL on research and new drug discovery in HBV. Has it taken a backseat in the COVID era? Let's address that. So today, I'll be able to answer that I don't think it has taken a backseat because there has been many new classes of antiviral agents being discovered and trialed for hepatitis B. What I've put for you here is a list of the current new molecules: capsid assembly modulators, or CAM, silencing RNA, or RNAi, antisense oligonucleotides, other oligonucleotide approaches. There's been some immunological approaches as well. We might have time for hepatitis B delta co-infection. And of course, because of the complexity of hepatitis B, combination treatments will probably be needed. Let's see how we go. Well, this slide shows the viral replication life cycle and potential targets as highlighted on the earlier slide. This is taken from a recent review written by myself and Fabian Zulem and Dr. French down below. And you can see here that the life cycle that most people are familiar with, the first class is the CPAMs, or the CAMs themselves. They act at the step of reverse transcription. The RNAi targeting drugs act at the level of the RNA coming out from cccDNA. And the third group, the NAPs, or the antisense stop compounds, they act inhibiting directly surface antigen release. Unfortunately, we don't have any drugs or antivirals targeting the cccDNA, and so unfortunately we won't have time to cover those. Let's first have a look at the first group of the antiviral agents, which are the capsid-associated inhibitors. And the first one that I wanted to highlight is, of the three major groups, is from Assembly Biosciences in America. And they have a second generation core inhibitor that's very potent. And as you can see from this data that was presented last year at AASLD, this capsid assembly modulator is very effective at inhibiting at least 3 logs of HBV DNA in a 2-week period, which is very, very potent. And the other key marker at the moment that we're all following in hepatitis B is the change in pregenomic RNA in the blood from baseline. And that usually tracks a little bit behind the viral load change. And here it's just on 2.5 to 3 logs. So it's a very active group of compounds. And I think we're going to see much interest in this over the next few years. The other company that's actively involved in capsid assembly modulators is Janssen, or J&J. They have a new generation of capsid assembly modulators. Their lead molecule here is 0440. And you can see here that, again, like in the previous slide from Assembly Bio, their drug is very active and very quickly produces a greater than 3 log reduction in HBV DNA. Similarly, 2 to 3 log reduction in RNA, and a more modest effect on the core-related antigen. So again, another series of potent capsid inhibitors coming from Janssen Company. The third group I'd like to mention is the GLS4, which is comes from a company in China, the Sunshine Lake Pharma, in collaboration with Peking University. And they've got an earlier generation capsid inhibitor that had PK and safety issues, and they've modified their treatment protocol to include a ritonavir boosting step, because these drugs are heavily metabolized by cytochrome P450 mediated metabolism. And they've changed the formulation of their compound and done a clinical trial, and the results are quite promising. So here you see the HBV DNA on this ritonavir boosted CAM is showing a good 2 to 3 log reduction, or even 4 log reduction in viral load. Not much effect on surface antigen or E. Oh, sorry. But also a potent effect on HBV pregenomic RNA, a 3 log effect. So what this teaches us is that drugs, even with unfavorable PK and PP pharmacodynamic issues, can actually be improved with boosting drugs like ritonavir to able to minimize the negative effect on the metabolism of the drug. There were significant flares observed in the high dose, and that will need to be followed up in the phase 2 study. Now, the next area that I'd like to touch on in this presentation is the targeting of the viral RNAs. There's been a lot of interest in silencing RNA, which is shown for you here, siRNA, and also antisense oligos. Now, the silencing RNAs, RNAi, gets metabolized or processed through the risk loading apparatus, and the risk loading apparatus is selective degradation of the viral RNA. In the case of the antisense oligonucleotides, the target is also viral RNA, and these are DNA molecules which bind to the HBV RNA, causing the hybrid DNA RNA hybrids to be metabolized by the enzyme RNase-H. And both of these are very effective antiviral agents, but delivery has always been a problem. And shown for you on panel B here is ways that chemists have actually modified these drugs, these RNA molecules, DNA molecules, to include GalNAc. And you put GalNAc on the end of the molecules, it makes it more likely to be taken up by the acylglycoprotein receptor, shown for you here in this slide as ASGPR. And this improved delivery is theoretically at least hoping to increase the uptake, processing, and delivery of these potent molecules to the hepatocyte. Excuse me. The first drug I'd like to mention in this is the collaboration between Arrowhead Pharmaceuticals that developed the Arrow-HBV TRiM platform, which they subsequently licensed to J&J Janssen. And so that is now the compound J&J 3989. And this is the first study showing a combination analysis of combined with a nucleoside analog in expanded cohorts of patients with chronic hepatitis B, doing a dose ranging study. And you can see here, these investigators looked at a number of different doses and got very good effects in terms of surface antigen reduction in the 200 to 400 milligram range, shown for you here in the lower right-hand corner. Very, very good, and heading in the right direction, declines in surface antigen. And because of the dose range initially studied, the company wanted to expand the cohorts to 100 to 400 milligram range to identify how low could they go. 25 and 50 milligrams were effective in reducing surface antigen, but appeared less effective than the higher doses. And the silencing RNA strategy seemed to be effective against both CCC DNA as well as the integrated DNA-derived HBV RNA. And overall the drug was well tolerated. It's given subcutaneously once a month for 3 months, and there was some very modest injection site changes which were adequately addressed. There were certainly no cases of ALT elevation greater than 3 times the upper limit of normal or bilirubin rises. The really excitement in the field at the moment is this study also from J&J Janssen, looking at the triple combination therapy of 3989, their RNAi drug from Arrowhead, J&J 6379, their in-house capsid assembly inhibitor, in the background of nucleotide analogs. So this was a longer-term study of this triple combination to explore the ability of the CAM active drug, a continuous dosing for this period, with the 3 monthly doses of 3989. And you can see the steady decline in surface antigen shown for you here very nicely, with the decrease down to almost 2 logs. So the conclusions from this study out to day 113 is that the surface antigen declined during treatment, a 1.4 log reduction by day 85, almost 2 log reduction by day 113. All patients at enrollment had more than 1,000 international units per mil of DNA, and that showed rapid decline, not surprisingly. The quantitative RNA, HBV RNA, also showed rapid decline of up to 7 logs. No serious events were reported, and there was a noted grade 1 rise in serum ALT in 5 patients. So this is promising to go forward, and I think we'll hear more news about this in the next few months. The other RNA active targeting drug is Antisense oligos, and this targets all HBV RNAs. And ISIS, in collaboration with GSK, have developed what they call 2-mo antisense oligonucleotide for increased stability, which shows a potent antiviral activity over a 28-day study. So these were a mixture of E-antigen positive and E-antigen negative patients, given 6 subcutaneous injections and followed on a background of tenofovir and tenofovir follow-up. And it was pretty well. There was a post-treatment flare in a single patient with the HBV and the surface antigen below the limit of detection. So the key viral markers are shown for you here. You can see that change from baseline in terms of surface antigen at the 2 doses explored, 150 milligrams, 300 milligrams, and the similar changes to HBV DNA. There were some post-treatment flares with delayed rebound of surface antigen in 3 E-antigen negative patients, which are being explored. But it gives promise for proof of concept that this approach could be quite a benefit. As I said, with these number of molecules available for treatment, it's now possible to consider and entertain possible other combinations. And this is a study from Roche looking at their locked nucleic acid for increased stability, antisense oligonucleotides, and a Tol-7 agonist in a mouse model. And what it shows is proof of concept. I thought I'd include this for viewers' interest. And you can see here that during treatment, there's a significant decrease in with the Roche Tol-7 agonist, and these both in terms of surface antigen, DNA, and with and without the antisense oligo. What was interesting in this study was as well as the controlled and viral markers, that there was a brisk B-cell response observed, and the view was that the Tol-7 agonist was the major driver of the antibody response. So this is a clear demonstration of the fact that a broad-based immune response is probably going to be needed in terms of achieving a functional cure. And a lot of interest was expressed in this presentation. The final group of compounds I want to look at is what they call the STOP group. These are like aptamers, if you like, the complex oligonucleotides. People might be familiar with the NAPs from Replicore. These are a new generation of aptamers called STOPs, which stands for Surface Antigen Traffic-Inhibiting Oligonucleotide Polymers. And this is an in vitro study which really looked very closely at the mechanistic basis for how the STOPs actually work, which is what the field needs. And you can see here is a Replicore equivalent, 1004, and it's much less potent than the current, these are from Aligos companies in America, a very potent dose-response effect in vitro. It's almost 2 orders of magnitude more potent than the current Replicore molecules, and shows a nice dose response. Here is a nice spread of the antiviral dose response effect compared to the Replicore compounds, which are a bit more of a cliff type effect. It's not what we want. So these compounds certainly inhibit surface antigen from multiple genotypes. They're reducing intracellular levels, which is very, very important. And they've had effects on other viral markers. One of the concerns about these compounds has been their ability to activate the immune response non-specifically. And the STOPs shown here had no immune activation in PBMC assays and showed synergy with the antisense oligos in reducing surface antigen. And they had good PK in terms of liver concentration. So I think we're going to see more of these aptamer type compounds in terms of their potency, good clinical dosing, no immune activation, and good PK. I don't think we'll have time for the delta, so I might move on from that. But just now to summarize, to answer the question of the APASL people asked me to address, there's been extensive research and new drug discovery in HBV at present in spite of the COVID-19 epidemic. I think the field is taken very good approaches in terms of developing COVID contingencies, and these seem to not be having a too adverse effect on the clinical trials. Novel antiviral targets have been identified, including direct-acting antivirals and viral RNA, and the adoption of liver targeting and improved PK/PD will, I think, broaden the armamentarium for hepatitis B. We've seen many examples now of combination studies being discussed between companies, which is good, and I think there is genuine optimism for functional cure in the future. Thank you very much for your time.